Okay. So welcome back after the coffee break. So it's my honor to present today the fifth session about microglia activation. And it's also, we have in this session two talks which will be online, so the speakers could not come, unfortunately. And so the first speaker is Andrea Ablasser. She's talking about cGAS, STING drives aging, related inflammation, and neurodegeneration. So
Okay.
Can you hear? Can you hear us, Andrea?
Yes.
Good,
Yes,
perfect. You can start the presentation.
Okay. So I guess you can see my screen and my slides. Yes. Okay, perfect. Yeah, first of all, thank you very much for the invitation. And I really apologize that I was unable to attend in person, but still I very much appreciate to have the chance to speak here online. So I am the lab, or the major focus of my lab is to understand nucleic acid-induced immunity, and we came across an interesting phenotype that I would speak about on neurodegeneration and microglial activation. But let me first introduce to you a little bit the topic and get you familiar with the mechanisms underlying nucleic acid immunity. Okay, so a principle in sensing viral infection that had been established decades ago is that foreign nucleic acids can trigger receptors and signaling cascades, which then drive a type 1 interferon response. And that's really a very fundamental mechanism of antiviral immunity. But one problem with this concept is that, as you all know, nucleic acids, they're not specific to viruses or pathogens in general, but in essence, they are the definition of life, and the host expresses a lot of nucleic acid, or contains a lot of nucleic acids. And so the question, and rather than this very simplified model that nucleic acids, or that specific foreign nucleic acids are being recognized, it rather appears that for this system to work, there are essential safeguards that prevent typically self-nucleic acids from driving these receptors and signaling cascades. However, as you can imagine, this might be a vulnerable system, and these safeguards during aging or disease might become perturbed or disrupted. And in those contexts, our own DNA or RNA might engage these receptors and drive innate immunity. And in certain contexts, as in for example, during tumor immunity, this can be beneficial. But in other cases, and I will show you one prominent example today, it drives immunopathology. So we were interested in understanding how DNA is being sensed from within the interior of a cell, and a pathway that had been discovered now about 10 years ago is the so-called cGAS pathway. Some of you may have heard of this. So it's a signaling cascade that relies on an enzyme called cGAS that produces a cyclic dinucleotide second messenger upon engaging double-stranded DNA released from viruses or bacteria, and that then the second messenger is a very potent activator of STING, and STING is a transmembrane protein that's expressed at the ER, and upon activation, it then traffics and engages at the Golgi kinase TBK1, then drives then downstream activity through engaging transcription factors, especially RF3 and NF-kappa B. And this pathway is most well known for driving type 1 interferon responses, but I should mention that this pathway also activates many other innate immune mediators and NF-kappa B targets. Now, it turns out this pathway is really important for sensing numerous pathogens, DNA viruses, RNA viruses, but those indirectly through the release of double-stranded DNA from the host. And so if you think about this pathway, you have a very unspecific sensor that senses a molecule which is abundant in host cells. On top of it, it's an enzyme which allows for multi-turnover production of a very, very potent immunostimulatory molecule. And on the other hand, a STING or binds to this cGAMP with very high affinity. It becomes clear that this pathway is really geared towards sensing DNA and translating this into a powerful immune response. But on the other hand, it's clear that this pathway requires regulation. And over the past years, many labs and our own lab contributed to identifying cellular safeguards that prevent sensing of self-DNA during or in the context of normal, healthy young cells. And so a cell invests a lot in degrading DNA that is, that emerges during normal DNA metabolism. There are important enzymes in the cytoplasm or in the lysosome, and if those are mutated or ablated, there is a very potent cGAS-independent auto-inflammatory response. Also, physical separation is important. So DNA is sequestered in the mitochondria or within the nucleus. And if nuclear DNA leaks into the cytoplasm or mitochondrial DNA, that triggers a type 1 interferon response through cGAS. And finally, we know also that accessibility is key. So DNA or chromatin factors can bind to DNA and prevent cGAS from sensing DNA. Another important mechanism that we and others more recently discovered is that cGAS is also present within the nucleus. And there it's very tightly tethered to the nucleosome surface. And this prevents it from engaging DNA because it's more tightly bound to the nucleosome surface. And just because this will be important later for my talk, if you mutate one of those arginine residues that anchor cGAS to the nucleosome surface, this triggers release from its inhibitory interaction with the nucleosome and drives cGAM and type 1 interferon responses. And this allows us to study in a very precise fashion consequences of DNA-induced cGAS activity. And finally, just to complete, cGAM can be degraded and secreted out of a cell to dampen cell intrinsic activation. And finally, there's a lot of regulation happening at the level of STING. STING is a very interesting molecule. It traffics, it only activates TBK1 at the Golgi, and then it's incorporated into clathrin-coated vesicles, which then are further delivered to the lysosome for degradation. And we've more recently shown that STING is actually encoded by clathrin-coated vesicles, which then mediate this transport to shut down signaling. So now you can maybe appreciate how important regulation is and how many steps are working together to typically prevent a very balanced, transient innate immune response to preferentially foreign double-stranded DNA. But this, as I mentioned, these safeguards can be overcome and drive inflammation. And there are many types of self-DNA species that have been associated with activating cGAS-STING in very different disease contexts: mitochondrial DNA, extracellular DNA from dying cells, endogenous retroelements, DNA damage byproducts, and so forth. And those have been linked into a very huge variety of distinct outcomes. As I mentioned, beneficially, it can be, for example, in natural or therapy-induced anti-tumor immunity, but many distinct diseases in mouse models, in preclinical models, but more and more also in human patients have been shown that aberrant DNA can drive a detrimental inflammatory response reliant on cGAS-STING signaling. And as you can see, there are multiple different types of diseases, and there's actually quite some neurodegenerative diseases that have been linked to this. Now, how can we explain this broad variety of diseases? Now, very simplified, you can think about out-of-context DNA being just one effect of loss of homeostasis, which is at the origin of each disease. And this out-of-context DNA can either drive acute or chronic inflammation that then by itself, the inflammation by itself is a driver of the disease. And with this framework, one can think about an intervention that blocks the collateral inflammatory response as a byproduct of loss of homeostasis, as a perhaps novel therapeutic intervention into chronic inflammatory diseases. And so some time ago, my lab has characterized first-in-class small molecules that can block STING allosterically, and they have been very useful in probing this pathway in different models of diseases. And so I will now talk about our more recent findings of STING and aging-associated inflammation. As you all know, subtle inflammatory states is a hallmark of aging and had been linked to various different causes, and one of which is cellular senescence. And some time ago, we and others have shown that the cGAS-STING pathway is engaged during the senescence response. And so this motivated us then to ask whether perhaps the cGAS-STING pathway might also be involved in aging-associated inflammation in an organism in vivo. And so this project was really headed by a very talented senior scientist, Fatih, and a very talented PhD students, Natasha, and then later Alex joined the project, another postdoc in the lab. And so just very briefly as a reminder and proof of concept, senescent cells that can be induced through various different treatments drive an inflammatory response. And once these cells reach their senescent state, and we treated them with the STING inhibitor H151, we could see that there was a very strong or a very significant reduction of the inflammatory cytokines in various different contexts of senescence. In this case, it's a radiation-induced senescence. Importantly, this senescence or the STING inhibition did not completely ablate the senescence response. Other hallmarks of senescence, such as growth restriction or beta-gal staining, were unaffected, so indicating that STING selectively affects the inflammatory response of these cells. And this was also confirmed independently by RNA sequencing. As you can see here, there's this characteristic inflammatory response that emerges upon once cells have reached the senescent stage, and that can be blocked by STING inhibition. We also showed as a proof of concept that this not only works in a petri dish with human cells, but also in human adipose tissue explants. And these explants accumulate senescent cells, as you can see here by this blue staining for beta-gal. And this is just a senolytic where you can see that these are senescent cells. And what we've, similar to the cellular assays that we did, we can see that these in primary senescent human cells, inhibition of STING dampens the inflammatory response of these senescent cells and further and improves their natural function. In this case, it's in the upregulation of genes involved in adipogenesis. But now let's move to the mouse model. So what this was our setup. We used aged mice and then treated them with a STING inhibitor and then assessed various different outcomes. And amongst the first is we profiled different organs for their inflammatory phenotype. And so this is an example of the kidney. And as you can see, as expected, we see this upregulation of inflammatory markers and also interferon-induced genes. And this could be blocked significantly, characteristically upregulated in aged mice. And aged mice that received H151 showed a reduction in these inflammatory cytokines. Importantly, we also aged a cohort of STING knockout mice. And we observed a very similar phenotype, indicating that really STING is one factor that is important for the inflammatory phenotype that develops during aging. Importantly, this was not only, or STING inhibitor treated mice not only showed a reduction in inflammatory cytokines, but they also showed improved organ function. And for example, this is for the kidney, shown regular kidney damage markers such as creatinine or urea were downregulated in the mice that received a STING inhibitor. And physical function also improved. So this is grip strength or treadmill performance. And you can see that the H151 treated mice did better than the vehicle treated control. And then finally, we also subjected them to cognitive function tests, especially like testing learning capacity with a fear conditioning test and spatial memory with a Morris water maze. And we saw a significant improvement in the mice that received H151. So indicating that STING inhibition reduces inflammatory features in the periphery and improves physical function and cognitive function. Importantly, this is a derivative of the H151 with better brain penetrance. Importantly, we didn't see any benefit in the overall survival of these mice, so indicating that STING doesn't affect lifespan, but when started at late in life, it improves health. Now we turned our attention to the brain because we thought this was a very interesting phenotype. And so we profiled by bulk RNA sequencing the hippocampus of these mice. And as you can see here, there's this immunoreactive signature that appears also in the brain of aged mice when compared to young mice. And importantly, that H151, the STING inhibitor, reduced also the inflammatory brain signature. I should mention that also H151 has brain penetrance. So we more carefully looked at other signs of neuroinflammation, and one of which is microglia activation. So there were more microglia during aging. That's hallmark of aging, actually known. But STING inhibitor treated mice showed a reduction in EBA1 positive cells. And importantly, they also showed reduction of GFAP immunoreactivity, indicating that there was reduced activation of microglia and astrocytes, and vice versa. There was less reduction in neurons in the hippocampus and less reduction of synaptophysin activity, indicating improved neuron viability. On the other, we again confirmed using the STING knockout a similar phenotype with reduction of activated microglia or microgliosis in the STING knockout and improved neuron survival. So how does it work? We first asked whether this effect on the brain is derived from a brain intrinsic activity of the cGAS pathway. And so the most specific biomarker of this pathway is measuring cGAS activity. And this is from aged mice, from brain lysis of aged mice. And as you can see here, there's increase of cGAS in aged mice compared to young mice, indicating that cGAS is produced within the brain. And that could also be confirmed by looking at phospho-STING. So this is a specific marker indicative of activated STING, so it becomes phosphorylated upon activation. And there was an increase of phospho-STING in aged mice, and most of this phospho-STING activity was recorded within EBA1 positive cells, indicating that STING is engaged within microglia in the aging brain. And we confirmed that ex vivo treatment of microglia with H151 indeed reduced the signature that's characteristic of aging, in such as interferon-induced genes, as shown here. So what's the trigger? What's the driver of cGAS during microglia aging? So one notable phenotype that we observed and that had actually also been reported before in the literature is that microglia accumulate mitochondria with a disrupted morphology. As you can see, they're blown up. They lack this characteristic and intact cristae structure. And as I told you before, mitochondrial DNA is a hallmark of, or is a major driver of cGAS-STING activity. And so we considered that perhaps mitochondrial DNA might engage cGAS in microglia. So to do so, we turned to super resolution microscopy of primary microglia. So these are microglia from young mice, stained for TOM20, which is an outer mitochondrial membrane protein, and double-stranded DNA. And as you can see here, most of these double-stranded DNA spots are contained within the mitochondria. However, in aged mice, what we observed, first of all, this very severe disrupted mitochondrial morphology that we observed also by transmission electron microscopy in situ. But furthermore, most of the DNA spots that we observed were located outside of the mitochondria. And this is just the quantification of those DNA spots. And we've also verified this by doing qPCR, where we do see upregulation of mitochondrial genes in aged mice. Importantly, when we block one of the major channels that had been implicated in mitochondrial DNA release, and these are VDAC channels with a small molecule inhibitor, we can reduce the inflammatory signature and type 1 interferon signature of these microglia cells. So what I've shown you so far is that in aged microglia, there is out-of-context DNA that drives cGAS-STING activation, promoting inflammation and neurodegeneration. But as you know, there are many other inflammatory, probably or many other pathways that might synergize with cGAS in the context of aged microglia. And so we were wondering whether this pathway might also be sufficient to, when once engaged, to drive an inflammaging similar or an inflammaging-like response in young microglia. And to do so, we turned to a genetic model that I've introduced that's based on a very important regulatory mechanism that I explained to you before. And this is that cGAS is inhibited by the nucleosome. Single point mutation can overcome this inhibition and drive a double-stranded DNA-dependent cGAM response. And just as a proof of concept, when we generated mice that express this allele upon tamoxifen treatment, and these are fibroblasts from some of these mice. You can see that upon tamoxifen treatment, there is cGAM production and there is interferon upregulation. So it's, to the best of my knowledge, the only genetic model in which we can genetically control DNA-induced cGAS activity. So we crossed these mice with free that's expressed from the TMEM119 promoter that allows us to express this mutant in microglia and brain macrophages. And when we did so, we could see that upon tamoxifen treatment, we did see microgliosis, so indicating that cGAS itself is sufficient to activate those microglia, and also by, and they recapitulated, cGAS activation recapitulated many hallmark features of activated microglia cells, as shown here in this 3D imaging. So these shortened dendrites and this activated morphology phenotype. Also, we observed upregulation of type 1 interferons and other inflammatory markers that had been linked to disease-associated microglia. And importantly, all this could be reduced by STING inhibitor treatment, confirming that these effects are dependent in STING, and again, independently proving that H151 is brain penetrant and can reduce inflammation in the brain. Now, I think I don't need to explain in front of this audience that very landmark studies a couple of years back have demonstrated that microglia should distinct, or that during disease states and also aging, a subset of microglia adopts a very characteristic transcriptional signature. And disease-associated microglia had been characterized also a similar overlapping neurodegenerative microglia subset, and also interferon signatures are going up in certain disease states in a subset of microglia. And so we were wondering whether cGAS activity would suffice to drive those transcriptional states and drive homeostatic microglia into either of those states. So we did single nuclear sequencing, and indeed could identify DAMs, interferons, and neurodegenerative microglia states. And what we first observed that upon engagement of cGAS, there was this overall increase of interference, also in the homeostatic microglia population, but you can clearly see this enrichment within the interferon subpopulation, so indicating that cGAS drives, enrichment of this subtype of microglia. And further, we also observed there was a further promoting of a damp state upon cGAS-STING activation, so indicating that cGAS on its own is sufficient to drive transcriptional states that had been identified in neurodegenerative diseases. We also profiled other cell types, oligodendrocytes and astrocytes, and what we observed that also within the oligodendrocytes, there was an upregulation of genes that's been associated with multiple CNS pathologies. And importantly, in this case, we engage cGAS only within the microglia, but it shows that this has bystander effects on other brain cells. Then the functional defects are shown here. So activation of cGAS not only changes the transcriptional status of microglia and their bystander cells, but it also had functional consequences. So engagement of cGAS reduced spatial memory capacity in the Morris-Water maze test, and also reduced neuron variability in the hippocampus. Now, finally, these are my two last slides. We were interested in identifying mechanisms of how cGAS drives or impacts neuronal survival. So we set up a co-culture system with microglia, primary microglia, and primary neurons. And upon engaging of cGAS with this tamoxifen-inducible system, as you can see here, there was reduction of neuron survival, allowing us to test, potential cytokines that might be involved in this phenotype. And so we blocked interferon receptor signaling because this was maybe our first guess that this might be a driver of neural toxicity. But in fact, as you can see here, blocking interferon signaling did not impact or did not rescue neuron survival, but instead TNF-alpha blockage did. And TNF-alpha was actually one of the highest induced cytokines in this model. And we've confirmed this also with aged microglia, so primary microglia collected from aged mice. Again, showing that rather than interferon, it seems that at least in this very simplified assay, TNF-alpha, which has been shown to exert neuron toxic effect, and plays a role in driving neuron inhibiting neuron survival. So with this, I'm coming to the conclusion. So there's many, as you know, there's a huge many diverse neurodegenerative diseases had been linked to inflammatory features. And you could have thought that maybe every disease might have its own pathway. But in the literature, it had already been reported that this pathway seems to be important in the context of models of ALS, models of Parkinson's disease, models of Alzheimer's disease. And so we now show that also during aging, the cGAS pathway might play a very important role. And so it seems that really the cGAS pathway seems to be an important general regulator during neurodegenerative disease. So with this, the paper had been recently published, and the cGAS pathway might fulfill one of these, or cGAS and STING might fulfill the definition of being an antagonistic pleiotropic gene product, which are very important for protecting us early in life from infection, which might become detrimental late in life. So with this, I'd like to thank all the people involved in my lab. I've shown the pictures before. I also want to thank Marco Prinz, our collaborator on this project, and funding agencies, and I'd be happy to take questions. Thank you.
So we have time for a couple of questions. Yeah.
Thanks very much. That's really incredible work. It's really great to hear that. I guess I've got a couple of questions. Do you think it's worthwhile doing chip-type experiments to try to identify what kinds of nucleic acids are being bound by STING, or do you think it's too promiscuous, it's binding too many species?
Yes, I think that's a practice that is being done, and I think in certain contexts it's really yielding, I think, very very interesting results. So mitochondrial DNA have been pulled down, genomic DNA can be pulled down, retroelemental DNA can be pulled down. So I think in general it is possible, it's feasible. It's just important to perhaps apply proper controls. It could, even so, maybe mitochondrial DNA plays a more prominent role in one context. We cannot exclude that, in very complex disease settings such as aging or cellular senescence, it might be a synergy of different types of DNA that fall into this pathway. And if you take one away, it might be sufficient to reduce the phenotype, but still maybe you can still find other DNA species bound in certain contexts of the disease or pathology.
Yeah, thanks. That makes a lot of sense. My second question relates to STING inhibition. Are there any negative effects with inhibiting STING in later life? You know, maybe infectious diseases?
Yeah. Yes, as I said, so the cGAS pathway is very important for, protecting against viral infection. And so, so any type of medicine that would block STING would have as a side effect immunosuppression. Now, that said, still the advantage of blocking one pathway instead of anti-cytokine therapy, for example, blocking interferon, blocking TNF, is that you leave intact all the other pathways that transduce viral infection. And we know that, even so in certain cell types, the cGAS pathway is essential for sensing viral infection, TLR9, or can compensate at the whole, at the level of the entire, of the whole organism. So, and their RNA pathway is intact and so forth. So as I said, it's probably comes with certain increased risk of infection, but if that can probably be controlled. And the other important consideration by thinking about a therapy is that we know that so the compound that we're using, this was the first compound that now they're much more progressed during clinical development and much more potent compounds, and we now know this. We achieve probably only 50% of inhibition of STING, but still we obtain measurable benefits. So that means that one thinking is that you probably need to ablate this pathway entirely to get have benefits in terms of disease outcome. So you might still maintain a certain level of intactness of this pathway in case of a very of viral infection.
Hi, Andrea. It's Svetlana Konenkova here. A fantastic talk, as always, beautiful stories. So it's very interesting that it's TNF alpha, at least in your cultures, that seems to drive neuronal death. So do you think we can block just the TNF alpha receptors, for example, to the same effect? Or do we need to go upstream? Or can we block, for example, an FKB, which probably is the driver of TNF alpha here?
Yeah, so I think that's an interesting question. So actually we were asked to maybe do this in the mouse, but it's very hard to block TNF in the brain, as you may know, because of the brain penetrance issue. Also, this where we see that TNF has a major role. This is very simplified experimental setup, meaning that we disregard in the system astrocytes, and maybe probably that astrocytes have been linked to be a very important indirect mediator, maybe also of microglia activation, be like other components of the brain, endothelial cells and so forth. So I'm not sure whether TNF might also hold true in vivo to be a driving force. With regards to FKB signaling, I still think that the ISGs and the interferon-induced signature might play a role, maybe not directly through cytokine release, but perhaps indirectly. And perhaps this phenotype might take a bit longer to take place that you might see in a culture. And this is largely because this type 1 interferon signature in the brain during aging, during different types of neurodegenerative diseases is really conserved between rodents and also humans. And also the microglia signature and interferon signature is clearly associated with neurodegeneration. So I'd rather think that TNF might be one example, but I still would, consider the interference as having an effect on neurons.
Okay, thank you. We have to move on. Applaud again. Thanks for the talk.